Go. Thank you. All right, ready to rock and roll? All right, so next topic. I promise you, I'm pretty sure, at least for my lectures, the rest of the block, nothing's going to be as dense as the last two we did. So if you can survive those two, and most of you have seen that stuff before anyway, so it's not too hard. If you haven't, it's a little harder. I've been getting a lot of questions, a lot of email questions, which is cool. Ask me as many as you want. If they're too in-depth, I might tell you to come see me, which you're welcome to as well. I'm starting to build up some appointments, so you may want to check ahead of time, but my office door is always open at least. Okay, so next we're going to talk about, at least this is the title, protein trafficking and degradation. I also moved the post-translational modifications into this lecture. They couldn't fit into the last one. I don't remember if I actually, yeah, I did put the post-translational modifications in the learning objectives as well. Okay, so this is actually a continuation of where we left off. We talked about translation, really just the basics of translation and how it works. But the next part of this process is the protein trafficking. Some proteins are actually synthesized and assembled on free cytoplasmic ribosomes, which is sort of the way we talked about it yesterday. We didn't really talk specifically about where it was happening, but it was happening in the cytoplasm. We just sort of had this messenger RNA floating there in a small ribosomal subunit, then the large one came in. And that is how some proteins are actually translated. And those include proteins that are going to stay in the cytoplasm, mitochondrial proteins, and nuclear proteins. Now, if they're going to go to mitochondria or the nucleus or anywhere else, they have to have specific localization signals. And those are pretty well characterized. We're not going to look at the actual signals themselves, but if a protein is supposed to go to a certain place, it's going to have a signal. If it's a cytoplasm, if it's translated in the cytoplasm and it doesn't have a signal, it stays in the cytoplasm. If it's translated in the next version of translation we're going to look at, where we go into the endoplasmic reticulum and it doesn't have a signal, it gets sent outside of the cell. So those are the two default pathways. So some other proteins begin translation on cytoplasmic ribosomes, and that's kind of what we got going on here. But those ribosomes are quickly targeted to the ER, and that's what we've got going on right here. So how do we target translation to the ER? Well, the proteins in this case, the very end terminus, the first part that gets translated, has a signal peptide. The signal peptide happens to be composed of hydrophobic amino acids. Okay, and that signal peptide actually interacts with an SRP, or signal recognition particle. Okay, it recognizes that signal peptide, binds to it, and there is a receptor on the endoplasmic reticulum, this SRP receptor, that recognizes when SRP is bound to a signal peptide. That directs translation in that ribosome to the ER and translation to continue into the lumen of the ER. As you can see here, now so this is kind of showing you a time lapse of what's going on. And once the ribosome is docked on the endoplasmic reticulum, when translation continues, that newly synthesized protein, as it's being synthesized, is coming into the lumen of the ER. This is going to include your secreted proteins, and we're going to look at a couple examples today: insulin and collagen. We're going to use those as two examples of how this process works. But also they include proteins inserted into the cell membrane, lysosomal enzymes, for example, and we'll show you a disease associated with this. So the proteins, also the proteins that go into the ER generally are going to undergo a lot of post-translational modifications as well. Now that's not necessarily the only place post-translational modifications can take place. Certainly you can modify proteins in the cytoplasm, but there are certain characteristic protein modifications, post-translational modifications that occur beginning in the ER and then through the Golgi. And I think you've discussed that sort of trafficking in a cell biology lecture. If you haven't, you certainly will. Now, here's a real brief slide. Just says chaperones. Molecular chaperones are proteins that assist other proteins to fold correctly, and that's as detailed as we're going to get. Those of you who have had more advanced cell biology or have done a little bit of reading, your heat shock proteins, those are chaperones. So there's some proteins that don't need any help to fold, but many of them do need help to fold correctly. And basically what happens is if you think of a protein and it's hundreds of amino acids long and you just kind of throw it into an environment and kind of let it fold, it's not necessarily going to go through the orderly process that it needs to. And really that's what chaperones do, is they kind of hold certain conformations so the next level of protein structure can form and so on. And they can also, as the heat shock name implies, if you get some kind of denaturization from heat or anything else, they can actually assist the proteins in folding back. At least in some cases, they can fold them back. Okay, so you also have other events that can happen in this protein trafficking. One is trimming. Trimming means we're going to move, and generally speaking, this really only occurs at the N terminus. The C terminus, we usually don't do any trimming, but at the N terminus, in eukaryotes, many of the proteins are actually trimmed. One of the things we talked about yesterday was translation always begins with the methionine, right? Well, that isn't always the N terminus of a mature protein because of this trimming. So you can actually make any amino acid potentially be the N terminus because of this trimming. One example of trimming that we alluded to in the earlier figure, and we're going to see in the examples with collagen and insulin, is cleavage of the signal peptide. So that signal peptide that directs proteins to be translated into the ER, that always gets cleaved off. Okay, once protein translation, once the translation is directed into the ER and it begins, there's an enzyme that's going to come in and actually remove that signal peptide. Another example of trimming, some proteins are actually synthesized as inactive precursors. Why would we want to do that? Well, we'll see today. Insulin is a signaling molecule. Obviously, you don't want that to be active all the time. But in this case, we have to actually cleave it to make a mature molecule. Collagen, there's a little bit of extra amino acids. Actually, in collagen, happens to be on both ends that actually have to be trimmed off to make the active form. But a good example also is proteolytic enzymes. So these are enzymes which are going to digest proteins. Well, you need to synthesize these as inactive precursors, because if you didn't, it would just start immediately digesting all the proteins wherever it's being synthesized. Okay, in the endoplasmic reticulum, for example, if it was something that was being sent outside of the cell, or in the Golgi. And when you think of proteolytic enzymes, something we'll see later on this block, something like digestive enzymes in the stomach, which are pretty brutal enzymes that just kind of degrade almost all proteins. What we can do is actually synthesize those as an inactive precursor, so it's got an extra tidbit on it that prevents that enzyme from being active, and then only when it gets to the correct place is that that tidbit actually cut off and activate the enzyme. Or lysosomal enzymes, another real good example. Some post-translational modifications that take place. You can have phosphorylation of enzymes or other proteins. In some cases, the phosphorylation activates enzymes. In some cases, it deactivates enzymes. And consequently, we also have dephosphorylation as well. So you can phosphorylate and dephosphorylate enzymes or other proteins. This occurs on the hydroxyl groups of the amino acids within a particular protein that actually have a hydroxyl group. So that's our serine, threonine, and tyrosine, right? And as we're going to see later on, when we look at metabolism, this process of phosphorylating or dephosphorylating is used to regulate an enzyme's activity. So you can turn on pathways or turn off pathways. There's other covalent modifications that can occur, glycosylation, for example. Now, I already mentioned glycosylation when we talked about amino acids, and that's the addition of sugar residues to certain amino acid residues. It's either if we're talking about O-linked glycosylation, it's the serine or threonine hydroxyl groups. Tyrosine, for whatever reason, doesn't participate. And the amide nitrogen of asparagine if it's N-linked. So one of the questions that I've gotten on email is, why doesn't glutamine participate? And the short answer is, I don't know why it doesn't participate. I don't know if anybody knows for certain. It's certainly, glutamine's a different size. So for whatever reason, and actually whether or not these things undergo glycosylation also depends on the amino acids near it. So it's not just any hydroxyl group in a protein is going to be glycosylated, or any amide nitrogen. It's only certain ones in a certain context. So the size apparently plays a role, and the enzymes that have evolved only use asparagine. These are associated with membrane proteins, lysosomal proteins, secreted proteins. So this modification actually occurs in the Golgi. So if you're going to undergo this type of modification, you're getting translated into the ER. Okay. Hydroxylation is another covalent modification. A good example, one we're going to see today, is proline and lysine and collagen are extensively hydroxylated. And in this case, this hydroxylation actually is important for the cross-linking. So collagen is going to form this triple helix, and in order for it to really be super stable, you're actually going to get some covalent bonds formed. And in some of those covalent bonds are using the hydroxyl group that we place post-translationally on these proline and lysine residues. So that means, so one thing you can ask is, well, what's going to happen if you don't do that? And we'll talk about that a little bit later. Another high-yield example of a post-translational modification is vitamin K-dependent carboxylation of blood clotting factors. So there happens to be a glutamate in there, so you can carboxylate these glutamate residues. And for these blood clotting factors, they actually have to undergo this modification in order to be an active blood clotting factor. Okay, and so what happens if you don't do that? Well, then one of the things, so what happens if so it's vitamin K dependent, what do you think is going to happen if you have a deficiency of vitamin K? We're going to talk about it next week, but you can potentially have trouble clotting your blood, therefore you potentially bleed to death. Usually it's not that severe with just vitamin K deficiency, but it could be. And so we'll talk a little bit about that. But you also use drugs that will target this pathway, as you will see in pharmacology, and later on when you talk about some cardiovascular stuff. If you've got somebody who's prone to blood clotting and you're trying to control that, you can actually interfere with this. You can actually attach lipid groups. So if you have a protein you want to be anchored into a membrane, certainly you can have hydrophobic amino acids that could do that, but you can also attach a lipid group to it, and that can also potentially anchor it into a membrane. We can actually acetylate proteins. We talked about that yesterday when we talked about potentially removing histone H1 from DNA by acetylating it, which kind of neutralized some of those positive charges. Therefore, it didn't bind as strongly to negatively charged DNA. Okay, so we're going to look at some examples here. The first example we're going to look at of post-translational modification in protein targeting is proteins that are designated for the lysosome. So any protein that's going to go into the lysosome is going to go into the endoplasmic reticulum to be translated. So it starts with that signal peptide, then you got that SRP receptor, gets translated into the ER, and then consequently goes through the Golgi and undergoes certain modifications. We do cleave off the signal peptide, for instance, but if I told you, if you're going to, so with this system, when you get translated into the ER, I mentioned earlier, but I want to mention it again, if you don't do any particular signal, and you don't tell it where it's supposed to go, by default, what ends up happening is it gets secreted out of the cell. No special signal means that's going outside of the cell. Okay? Just like with if it's translated in the cytoplasm, if there's no special signal, it stays in the cytoplasm. So if you have enzymes that are supposed to be targeted to the lysosome, for example, the special signal that they contain is a mannose that gets phosphorylated. So this mannose is an example of a sugar. So it's a glycosylation event that occurs in the Golgi apparatus. And furthermore, beyond just that glycosylation, you actually have to phosphorylate that mannose. That is a signal that, okay, this enzyme is supposed to go to the lysosome. You guys know what lysosomes do, right? You already talked about lysosomes, right? Okay. So you know that they're basically, I like to think of lysosomes as the stomach of the cell. So they digest all different types of macromolecules. Okay, so we have this disease, and here's one example of where there is some stuff in the figure that I didn't write out here. I do want you to know eye cell disease, and it's just giving you a little tidbit about eye cell disease there. So I want you to know that little tidbit, okay, which I'll talk to you about. But I didn't specifically write it out, so that is an extra detail in the slide that I do want you to know. So what happens in eye cell disease is there's a defect in an enzyme, or we're not going to talk about the specific enzyme, but there's a defect in the enzyme which phosphorylates this mannose. Okay, so the signal that's telling these nasty lysosomal enzymes that they're supposed to go to the lysosome is missing. Okay, so if we don't have the signal, then we're going to follow the default pathway. And what is the default pathway? I just mentioned. Getting sent outside the cell, right? So in eye cell disease, we've got a couple issues here. First of all, we have this signal that's supposed to tell enzymes that they're supposed to go to the lysosome. That signal is missing. Therefore, all these things that are in the lysosome that are supposed to get digested, the enzymes that are supposed to digest them are not there. That causes the lysosomes to accumulate all kinds of garbage in there, and they actually, and literally that's where eye cell disease got its name from. It causes these inclusions in the cell where these lysosomes just get enormous. It is an example of a lysosomal storage disease. You're going to see a whole bunch of those because they're very high-yield diseases on board exams. Even though they're relatively rare diseases, they're very high yield on board exams. And so you get this accumulation of things in the lysosome. That itself causes a problem, just like with all the other lysosomal storage diseases. You get things that are supposed to be degraded in the lysosome. They don't get degraded. They accumulate. and they cause some types of problems, and it depends on what tissue we're talking about. But we've got another problem here as well with I-cell disease. Because we're not sending the enzymes to the lysosome, but we're sending them outside the cell, now all these nasty enzymes that are normally in the lysosome that are degrading proteins and fats and sugar and all these leftover components are actually being sent outside of the cell. Okay? And now outside of the cell is not an ideal environment for those enzymes. The lysosome tends to be a little bit acidic, so it's not the optimal pH for the enzymes, but they still have some activity. And so they start trying to do whatever their enzymatic activity is outside of the cell. And that contributes to the symptoms. So you get these cells that are misshapen because of the accumulation of things in the lysosomes, which causes issues. And then all these nasty degrading enzymes that get sent outside of the cell cause issues. And you end up with these symptoms, the skeletal abnormalities, restricted joint movement. I think you can understand how that's going to happen. Okay, coarse facial features. You get some neurological damage as well. And this is a severe disease, so death usually occurs by 8 years old. So once again, I-cell disease, even though I didn't write the stuff here, I want you to know, be familiar with I-cell disease. Okay, so we're going to briefly look at protein degradation and the two examples of protein degradation, or the two major classes of how we degrade proteins. One of them we just alluded to. So individual proteins do have distinctive half-lives that are related to their function. So if you have something that's supposed to be very short-acting, and we're going to contrast because I'm going to show you both of these today, insulin and collagen. These are on the two opposite extremes. Insulin is a signaling molecule. That means it's something you want to turn over very rapidly. That's the same with anything that's going to be a signal. You don't want the signal constantly on, or else you kind of defeat the purpose of even having a signaling pathway. Okay, so the signal has to go off at some point. And so insulin has a half-life of about 5 minutes. That's one of the shortest half-lives of any protein you will ever see. Of course, when it's finally released into the bloodstream. When it's first synthesized, it's going to be kept in some vesicles, and in there it's stable, but once it goes into the bloodstream, it's going to be turned over rapidly. While collagen, which is actually a structural protein, so that's something you want to last a long time, has a half-life that's on the order of years. Those are the two extremes, and most proteins are going to be somewhere in between there. Most of the proteins that we're going to talk about in biochemistry and all these enzymatic pathways, their half-life might be on the order of days to months, okay, depending on which pathway they're involved with. If you do have a protein that does get misfolded, and that absolutely happens all the time, so a certain percentage of all proteins get misfolded, they're going to be unstable and turned over. So your body recognizes proteins that are misfolded. It tags them for destruction, and we're going to talk about how it actually does that here briefly in 1 second. So this actually contributes to the pathology of some diseases. And I said the recently discussed, that's because previously I actually discussed this disease ahead of this. We haven't discussed glucose 6-phosphate dehydrogenase deficiency yet, so don't worry about that. But there are diseases where the mutation that you see in the protein actually causes a stability issue, okay? Actually causes the protein because it's less stable, it can assume an incorrect conformation relatively easy, and when it does, it gets turned over. So what ends up happening is those enzymes get turned over much more rapidly, and that contributes to the disease. This is a disease I will talk to you about, so we'll revisit this when we talk about the HMP pathway a little bit later on. So that's an example of where you could have a protein, you can have proteins which they enzymatically are perfectly fine, but they're unstable and they misform and they get turned over more rapidly, and that ends up causing disease. So the half-life of a protein is determined by its overall structure, and also you can have particular amino acids within a particular stretch of amino acids within a protein may target it for more or less rapid degradation. And a famous example is this PEST sequence. Now, in this case, I used for this, and it makes a very convenient name, PEST. We use the single letter abbreviations for the amino acids, which I told you don't have to try to memorize, but they're given to you there. But that PEST sequence is found in proteins that are supposed to be turned over relatively rapidly. Okay. Another thing we know about protein degradation is the amino acid at the N-terminus has a large influence on how quickly a protein is turned over. Now, when we looked at translation, you know, we always started with methionine, but now, because of the beginning of this lecture, you realize that methionine isn't always at the N-terminus. It could be if you didn't do any trimming at all, but any amino acid could end up at the N-terminus. Okay, and that will influence how rapidly that protein gets turned over. And we're not going to go through the specifics of which ones target it for a long-lasting protein or a short-lasting one, but just realize that that can happen. Okay, so lysosomes, which we already looked at, and I think you've looked at in some cell biology lectures as well. They primarily digest extracellular proteins and cell membrane surface proteins. So anything outside the cell comes in a vesicle, gets transported into the lysosome to be degraded. Anything on any of the membranes that need to be turned over gets sent to the lysosome. But intracellular proteins, that will include our misfolded proteins or proteins that are part of natural turnover. Eventually they will misfold, or eventually they'll get damaged and they need to be turned over. They're going to be digested by proteasomes, which is shown right here, kind of a visualization of what it looks like. So proteins that are actually going to be degraded by proteasomes are actually tagged by ubiquitin. Okay? And actually it's multiple molecules, as you can see in here, of ubiquitin. Now, ubiquitin signaling, if you've taken a really advanced cell biology class before, you might realize there's a little more to ubiquitin signaling than just protein degradation, but that's all we're going to talk about, okay? So I should say that's all I'm going to talk about. So one of the things that ubiquitin tagging does is acts as a signal saying this protein is damaged, this protein needs to be degraded, this protein is old, something's wrong with it, didn't fold correctly, we need to turn it over. Proteasome recognizes that and will actually digest that protein. So this is another way of turning over proteins. So you have your lysosomes and your proteasomes. Okay, so we're going to look at a couple example molecules. Kind of summarize a lot of what we've talked about over the last few days, and including some of what we've talked about today. The first one we're going to look at is insulin. Both of these are big molecules in medical biochemistry. Okay, so when we synthesize insulin actually originally, what's first translated is a much bigger protein than the final molecule of insulin. If you look right here, here's what insulin looks like at the end. It's actually got two separate peptide sequences that are joined by disulfide bonds. Okay, so it's kind of weird in that originally it's translated as a single peptide, but it ends up being two peptides joined by disulfide bonds. So how the heck does that happen? Okay, originally this is one that's going to be targeted to the ER. So what's it going to have if it's targeted to the ER? It's going to have that signal sequence, right? That signal peptide. Okay, that hydrophobic sequence, SRP recognizes it, binds to it. You have the SRP receptor. It's translated into the ER. What I tell you is going to happen to that signal peptide or that signal sequence after it goes into the ER? chop that off, right? So that's one of the events that's going to happen with insulin. We're going to remove that signal sequence. That gets chopped off, okay? Now, as insulin moves from the ER through the Golgi, you're going to have other processing events that are going to happen as well. You're going to have disulfide bond formation, okay? So what ends up happening here, and this is how we're going to end up with a dipeptide at the end. The insulin molecule actually folds back on itself, so that N-terminus and that C-terminus fold back on itself, and you have an oxidation event which creates these disulfide bonds. It kind of joins it initially as a loop. It kind of makes like a ring that's been sealed by those disulfide bonds. But ultimately, we're going to be left with a molecule that looks like this. So the next event that actually happens is we actually are going to cleave out a section in the middle, and it's actually a very substantial section of that original protein. So the final molecule of insulin, which looks what's right here, is actually, for a protein, that's actually very, very small. So sometimes people won't even call it a protein. They'll call it a peptide because it's got so few amino acid residues. So more of the protein is actually removed than ends up in the final protein. So this next section that gets removed is called the C-peptide. And it does play a role in medicine that we're going to talk about. So whether it has a physiological function is still being debated, but it has a role. Okay, so here's how things happen with insulin. First of all, just like with any gene, we have transcription occurring in the nucleus, right? We talked about that yesterday. That mRNA has to undergo its processing events, right? We get this 5' cap, 3' poly A tail. I don't remember if insulin has any introns in it or not, but if they are in there in the primary transcript, you have to cleave them out, right? So once all these processing events occur, you transport that mRNA out of the nucleus, begins translation in the cytoplasm. The very first thing, the very N-terminus, the first thing that comes out during translation happens to have that signal peptide, okay? So you're going to get binding to SRP and that SRP receptor on the ER, and translation gets sent, that protein gets sent into the lumen of the ER and then undergoes these processing events. That includes cleavage of that signal peptide, the formation of disulfide bonds, and the cleavage of this C-peptide to yield the mature molecule. Ultimately, the insulin molecule is going to follow the secretory pathway and be sent outside of the cell. You can store insulin for a period of time, so you can block. You don't have to immediately, when you first synthesize it, send it outside the cell. So it will actually remain in those pancreatic beta cells. It will actually remain as vesicles in there until the appropriate signal comes to finish going through that pathway. And you'll notice here, both insulin and the C-peptide get sent out, okay? So insulin is actually the signaling molecule, but the C-peptide gets sent out as well. So what's going on with the C-peptide? We're going to talk more than you ever wanted to know about insulin throughout the rest of biochemistry, but we're really not going to talk about C-peptide again other than today. So I'm going to save the story for all the things insulin does, which you may know or may not know, but by the end of this class you will certainly be familiar with this. It influences all the metabolic pathways we're going to talk about. So what does the C-peptide do? Well, for one thing, it's thought that the C-peptide really doesn't do anything. But it'd be kind of weird for something to be produced that just doesn't do anything. So it may have some functions that we're not aware of yet or are not 100% clear yet. But you could certainly do without it, because type 1 diabetics who are relying on insulin, that synthetic insulin they get doesn't have C-peptide in it, okay? And they're relatively normal. They certainly can live a normal lifespan. Now, there are complications of type 1 diabetes, even if it's treated, but As far as we know, none of those have to do with the absence of C-peptide. But you'll notice, because of the way translation works and that processing event works, for every molecule of insulin you send out, you also send out a molecule of C-peptide. So they're sent out in equal molar amounts. However, for whatever reason, that C-peptide has a much longer half-life. The half-life is on about 35 minutes, which is almost an order of magnitude bigger than the half-life of an insulin molecule. That means, ultimately, the circulating concentration of that C-peptide are about 5 to 10 times higher than the circulating concentration of insulin. What that ultimately means is it's much easier to detect C-peptide. So sometimes, some tests, when they're trying to test for insulin, is actually an indirect test where they're really measuring the C-peptide because it's easier to detect. So it's an indirect test. You can use math and work backwards and figure out what the amount of insulin should be if you know the amount of C-peptide, okay? So that's one thing that C-peptide is used for in medicine. Another thing you can use C-peptide for is, let's say we have a type 2 diabetic. If you're not familiar with type 1 and type 2 diabetes yet, don't get all frantic and feel like you've got to go home and study all that. We'll get to it eventually. But a type 2 diabetic is one they would call a non-insulin dependent diabetic, okay? Usually associated with obesity, and it happens a little bit later in life. Eventually, type 2 diabetes can get bad enough where you have to give insulin. Usually at first they try to control it through other drugs and some lifestyle modifications. So if you lose weight, sometimes that can help. But eventually you may have to use insulin. But one of the dangers of insulin, if you didn't already know this, you give somebody insulin, you give them too much, it can absolutely kill you, okay? Insulin will drop your blood sugar, and if you drop your blood sugar enough, it will kill you, okay? So insulin can be dangerous, and it's something you have to be very careful with. So let's say we had a type 2 diabetic. They're producing some insulin, but they're apparently not producing enough, or you thought they weren't producing any anymore, and you prescribed insulin, and then they come in the emergency room, they OD'd on insulin, and you're trying to figure out, okay, are they still producing insulin, and the test we did earlier was inaccurate? Well, one of the things you can do to differentiate between endogenous and exogenous insulin, so the insulin you inject in type 1 diabetics or in extreme cases of type 2 diabetes doesn't have that C-peptide. So only the endogenously produced insulin does. So in this case, you can look for how much C-peptide is there and then figure out, okay, how much of the insulin came from the injection and how much of the insulin was actually residual activity from the pancreas, okay? Next thing we're going to look at is collagen. Okay, once again, this figure kind of summarizes a lot of the processes that we've already looked at. Obviously, you have to begin the synthesis of collagen with transcription, go through all those processing events on the messenger RNA, transport it out of the nucleus. It's going to have to be translated. This is one that gets translated into the ER, so it's also going to have a signal peptide. Signal peptide actually has to be removed. Now, you'll see I use some abbreviations here. This OI, you already know what that one is, right? Osteogenesis imperfecta. EDS, I'll explain in a second if you haven't seen it yet. Ehlers-Danlos syndrome, scurvy. These are some diseases we're going to talk about. And all I'm saying is these are possible areas where that disease is affected. And so collagen's going to undergo a lot of post-translational modifications, and I alluded to this earlier. It undergoes hydroxylation events. Okay, ultimately, these hydroxylations are going to be important to form the stable triple helix. It also gets glycosylated in there. There is sugar residues in there. You get disulfide bonds being formed, and you can see some of them there. It is a protein that has multiple polypeptides, so it does have quaternary structure. It's an example of one with quaternary structure. It is following the secretory pathway. It is supposed to be sent outside of the cell, so that means it doesn't have any special targeting sequence. It just follows the default pathway. And we do have some cleavage events where we're actually going to cleave off the ends of the protein, and then ultimately to perform, or I'm sorry, in order to make a mature collagen molecule, we actually do some cross-linking events that actually will link that triple helix and allow it to form a very stable, covalently bonded structure. So we're going to look at a few disorders of collagen biosynthesis, and I want you to compare and contrast these by the time we're done. Okay. One of the things I want you to do, and I didn't want to just repeat what I've already talked about, so I didn't include the slides here, and I've talked about it in 2 different lectures, but add osteogenesis imperfecta into your collagen disorder list. It is certainly a collagen disorder, and so I want you to compare and contrast that one with the ones we're going to talk about today as well. Okay? It's something you guys are probably already pretty familiar with, but I want you to. So, for example, on an exam, I might give you something, and you need to figure out which one of the diseases it is. Okay, so that's why I want you to compare and contrast and add OI in there. First one we're going to look at is scurvy. Okay, scurvy is not a genetic disease. Okay, scurvy can happen to anybody. Scurvy is a deficiency of vitamin C. Okay, vitamin C is actually a required cofactor for the enzymes involved in hydroxylating these proline and lysine residues in collagen. That means if you have a deficiency of vitamin C, it affects your ability to actually do this hydroxylation event, which ultimately affects the structure of collagen. And so in vitamin C deficiency, the symptoms you see because of the defects in collagen are easy bruising and bleeding. So it affects the structure of some of the tissues. In particular, you really see it where it first shows up. Ultimately, scurvy can get bad enough where it starts affecting things like bones. In particular, if it happens in a child that's still growing. But where you first usually see the symptoms and where it's first noticed is in the gums in the mouth are particularly sensitive, and they start bleeding. And you'll notice bruises in arms and legs and things like that. Somebody will bruise very easily. And also the wounds won't heal very quickly, because in order for part of wound healing is formation of new collagen. Okay, so here's an example of a non-genetic disease. The other ones we're going to talk about are genetic diseases. The next one is Ehlers-Danlos syndrome. You guys talked about that? No, I don't think so. Okay, so Ehlers-Danlos syndrome. Years ago, my original mentor, he said Danlos sounds like damn loose, so kind of visualize what's going on in Ehlers-Danlos syndrome, because you're going to see hypermobile joints and some things like that, so things that are loose. Ehlers-Danlos syndrome is an example of a genetic disease where you have multiple mutation types. Therefore, when I say multiple genetics, I mean multiple different ways of being inherited. These are things that could be autosomal recessive, dominant, X-linked. If you haven't seen those terms yet, don't panic. We're going to see those later on, and we'll revisit the story of Ehlers-Danlos. But most genetic diseases are very particular. It's a very specific enzyme, and in many cases, like sickle cell, for example, it's a very particular mutation. Ehlers-Danlos is, the syndrome is named clinically, so it's named based on the symptoms, not necessarily the molecular mutation. So there's multiple different things that could cause that. There are mutations that can happen in collagen genes that will cause the symptoms of Ehlers-Danlos. Remember, there's multiple different types of collagen, With OI, we were looking at type 1 collagen, but there's multiple different types of collagen. You could have collagen processing enzymes. For example, a defect in lysyl hydroxylase can cause Ehlers-Danlos syndrome. Or collagen peptidase can cause Ehlers-Danlos syndrome. So multiple different types of mutations. What are the symptoms of Ehlers-Danlos syndrome? Hyperextensible skin and hypermobile joints, which I'll show you a picture of in 1 second. You could also get easy bleeding and bruising, just like with scurvy. Okay, but scurvy is not going to have these hypermobile joints and hypermobile skin. But really, the big issue with Ehlers-Danlos, the issue that can be life-threatening, is the most severe forms of Ehlers-Danlos have potentially lethal vascular problems. So the collagen that's lending some structural rigidity to the vascular tissue is compromised, and therefore it can actually stretch out and cause an aneurysm. So that defective collagen in the arteries can be a potentially life-threatening issue. Here's an example of the hypermobile joints. There are other things that can cause hypermobile joints, by the way. So Ehlers-Danlos syndrome is one. Anybody in the class can do that? Usually we find some people that can do it. Can you do it? Oh, do you? Oh, interesting. Can you do the skin stretchy thing yet? Oh, okay. Okay. Yeah, so there's actually a whole bunch of different types of EDS with different, so not all types of EDS get the stretchy skin where they can pull the sugar out of their face, but I call those the fun symptoms of EDS, right? Because you can amaze your friends and stuff. But anyway, we'll revisit EDS when we talk about, when we get into genetics. Okay, another disorder of collagen biosynthesis, another disease that could end up on your board exam is Menkes disease, also called Menkes kinky hair disease. I've also seen it just called kinky hair disease because kinky hair is one of the symptoms of this. So in this case, there's a mutation in a copper efflux protein, and what it ends up doing is resulting in a functional copper deficiency. One of the enzymes that requires copper, you'll see copper is actually also required in the electron transport chain. We'll see that later on. But, and so that can cause some of the symptoms of Menkes. But copper is also required for the normal processing of collagen. Lysyl oxidase, and I don't know if we showed it in this figure, but anyway, lysyl oxidase has a copper requirement. And this enzyme, what it does is cross-link collagen fibrils. So what's going to happen? You're not going to be able to cross-link it. And then that results in the symptoms you see of Menkes disease, including that depigmented, so it actually looks almost like gray hair, and it's very kinky. So you could actually type this into Google Images and pull up. Now, this disease kills patients very early. They can try to do copper supplementation, and it helps a little bit, but really, so most of the pictures you find are of very small infants, so sometimes they don't have a ton of hair there, but you could probably find some good pictures there of what I'm talking about. But the real issues here are the effects of collagen on other things, with arterial ruptures, which are going to cause this cerebral degeneration. You get effects on the bone as well, osteoporosis, and therefore, because of the effects on the bone, affects bone marrow, the production of red blood cells, so you get an anemia. And this is a real severe disease, so this will kill within the first decade of life, and usually even within a year or two of birth. So this one is pretty severe. Once again, you could do some copper supplementation, and it helps a little bit, and can extend life, maybe to the first decade. But it's definitely absolutely not curative. Okay, so that's all we have for this lecture, and I will see you guys in a few minutes. Yes.
I just had a quick question about when you said you want us to compare and contrast. Do you want us to more know the symptoms or like the steps in the synthesis?
So with any disease in biochemistry, any disease that I talk about, this will be a rule from now on. If I mention an enzyme, I want you to know the enzyme. So biochemistry is really concerned with, if you're tested on a board exam, they're going to ask you about the enzyme one way or another. So if I give you an enzyme, I want you to know that, or I want you to know the molecular mechanism if I talked about it. Like with vitamin C deficiency, I want you to know why that causes the symptoms. And then for each of them, if I talked about some of the basic symptoms, in most of the cases, I tried to use logic to explain why that defect causes that disease. In some cases we don't know why. And so you'll see these random symptoms, and I'm like, I just don't know why it does that. We haven't figured that part out yet. But for all the diseases, you need to know the symptoms ultimately anyway as you go through for your board exam. So I'm going to ask you to go ahead now and learn the symptoms that I give you. In almost all these diseases, you're going to expand upon the story later on. But try as much as possible to use logic to explain the symptoms, because then it's not like memorizing. Then it's understanding. Okay? Does that help figure out? Okay. All right, see you guys in a couple minutes.